I'm at Bach Tower with my wife, partner, and we're going to be walking around with Cheryl Peterson. In fact, I'm shooting B-roll right now in my introduction. I've already done the walkthrough with Cheryl, so I like to do that because then I know what happened and I can sort of, rather than just guessing what you're going to experience, we go through, there's a greenhouse facility down there, there are bed gardens down here. We're going to look at some species in Lamiaceae, which are the Conrandina and the Dicerandra, and also the native jujube. There's a lot of work being done there, as well as a few other things. There's the Crotalaria avenensis, the, I've already forgotten the genus, but it's the Hillsborough aster. And so there's a bunch of stuff going on over there. And so let's get going. I'll string this together in sequence, and hopefully you guys will enjoy this. It'll be in multiple chapters, too.
Conservation has, we have our seed, it was an office, and now we have it for volunteers, some office-y type work, but it's mostly our seed processing facility. I'll show you that. We'll walk down here first.
Okay. Well, the tower is lighting up.
The supplies that you see over at Hallstrom, we grab them from here.
Right, okay.
And go, and as well as other processing supplies. And this is all nursery over here. That's the nursery manager's office. I haven't seen her today.
Okay.
I don't know where she is. Today. We have some supplies here, some seed drying cages.
Oh.
We have a really rare plant called Florida ziziphus that was once thought to be extinct.
Mm-hmm.
And that's sort of like when we give any talks about what our program is all about and why it's important. Is one of our poster child plants that we talk about. It's been studied by us and Archibald and a little bit by UF as well. And it was, rediscovered by like a handful of very small populations, like 2 to 10 plants.
Okay.
But those are all private lands that are going to be developed, or something, bad was happening to them all. And so we weren't able to take cuttings. That doesn't work. The roots are very extensive. There's no way to transplant a plant. And it produced loads of wonderful flowers, but never produced seeds.
Okay.
So it was like, well, you rediscover it just to kind of watch it go extinct again. So all these studies, we ended up learning that each of the populations were different genotypes and also mating types, and it's an obligate outcrosser. And in one population, they were all the same genotype, so they could never produce seeds. And in a lot of cases, what looked like 2 to 10 plants was really just one plant.
Okay, rhizomially, yeah.
Exactly. They form like the shallow root runners and come up as other ramets. So it's
okay,
that's really just one plant. So what do you do? But we ended up, my predecessors sort of ended up taking advantage. You need to go here?
My bad.
Needed to take advantage of its propensity to form the shallow root runners. So they took cuttings of the like pencil diameter roots that would come up and bring it in the greenhouse. So there'd be just like trays of dirt with a dead looking stick in it for maybe a year, and then you'd see it start possibly, it's like maybe about a 1% success rate. But eventually, if the lucky cuttings would form shoots and then roots, and then you get a clone of them.
Right.
So a lot of the plants that ended up in the National Collection here are clones from all the populations. To this day, there's genotypes that we cannot get in because it's so difficult to get them in the collection. But we have most of the genotypes in here, and so when those plants gradually grew to be big enough to be reproductive and form flowers, for the first time ever, that we know of, these guys could freely cross. And there's loads of pollinators. Pollinators love these things. And it just finished flowering actually in January. The flowers are amazing. And so they freely cross here. And so in about May, we'll start getting the fruit, which are like great big drupes that look like mini mangoes, and they actually taste kind of nice. They're very fragrant.
Reminiscent of a true jujube?
Yeah, exactly. And And so those seeds are really precious, since this is the only place in the world, effectively, that seeds are
That has
produced.
that, yeah.
But if the seeds are just left to drop when they're ripe, all sorts of predators get to them. And also, like when we collect them, since it has a large amount of pulp around it, we can't just like store them; it'll rot. You know?
Right.
So we've tried different ways, tried to remove the pulp, and the best way that retains the viability that we found is to lay out the seeds on drying racks until like it dries down and mummifies, and then they store well and they germinate fine.
Okay.
But we need to keep them in covered cages, and that's what all these are for. We just have these.
Okay. So you have pseudosisyphus in here right now?
No.
Oh,
no,
this, okay, but that in concept.
Okay.
Okay. Oh, that's right. Yeah, you were just saying that. Okay. So they are traditionally orthodox seeds?
Yeah.
Okay.
Yeah.
As opposed to
And we
recalcitrant.
had a volunteer make these, which is wonderful.
Oh, that's neat.
And so, we sometimes some plants have just a few seeds, and some others have a lot,
Okay.
and we could just
You can section out
Yeah,
as a...
so we fit like maybe 4,000 seeds into all of this.
Oh, wow.
And so as they dry, so we have 4 of these racks. We'll kind of pull this out, and just every day, every morning we go, during May and June, we'll go and like see what's ripe. What seeds are ready to come out, and collect them and put them in here, and then as they dry, then we could take them out and set up germination trials and process the rest for storage. But we need to keep them in covered cages, because we've noticed that if we just try to like dry them out in the open and in the greenhouse, like what we used to do, sometimes like our viability ended up being lower. And then we found like a little larvae sometimes coming out of, and it turned out to be a pink scavenger moth.
Okay.
At night they'll come in the greenhouse and anywhere else. They even find their way in here and lay their eggs just right on the cotyledon. The larva eats the cotyledon, and then a little hole and comes out. And so we lost a lot of seeds like that.
I can imagine.
This is important for
Yes.
the physical. So,
yeah, absolutely.
yeah. So.